Thank you. Hopefully, everyone online can hear me okay. We have a wonderful audience here in the auditorium. But yeah, my name is Valentina. I currently work at NHOBI over in Bethesda. I'm one of the two co-directors of the EM core facility over there. But today I'm going to be showing you work that I did over at UConn several years ago while I was still a graduate student. And it's a little bit different flavor from what Dr. Westlake just presented. This is going to be mostly to show you an introduction on the method, which is using the automated tape collecting ultramicrotome or the ATUMtome as a way of doing array tomography. So, okay. So if you're not familiar with the array of volume-EM methods, you will become familiar with them over the course of these 2 days. We're probably going to touch on a lot of these, but array tomography occupies this very nice niche over here, shown in green in this beautiful review paper, which I encourage. There's probably several authors from this in the audience right now. But what I want to highlight here is the versatility of array tomography, which is it allows you to image structures as small as, for example, organelles within cells, or as large as whole organisms in the case of C. elegans or something around that size. Now, some of the limitations do come from the resolution, especially the Z resolution, because you're physically sectioning your sample. You have a limit in how thin you can section that. So if you're a very good ultramicrotomist, you can probably go down to 30 nanometers per section. It's quite difficult, but if you are able to do that, the data quality improves dramatically. Otherwise, resolution per section, 60, 70 nanometers is also enough, and I'm going to show you a little bit of that data as we move forward. So what is the ATUM or ATUMtome as it's called now? So we start with your typical resin embedded tissue stained with heavy metals. This is put onto an ultramicrotome with your typical diamond knife with the diamond blade here. And here is the ATUMtome. So in this case, I'm showing you the prototype. This is kind of like a homemade version, which we got from the Harvard lab that developed this several years ago. But it's very simple. So what we have here is a tape, it's Captain tape. This is the intake reel. So what the tape does is it gets reeled through these two motors. So it's going to be moving this way. Then it moves up here. This region we'll call the snout. And this is what is actually inserted into the diamond knife underneath the water. And this is where the sections will be collected. Now the tape continues to go up this way. By this point, the sections are mostly dry. They go through here and then they're collected onto this final reel. And then all of your sections on tape are collected here. You can collect up to thousands of sections, really. There's virtually no limit as to how many sections you can collect, except how far your microtome distance goes. So sometimes you have to, it reaches that distance and you have to rewind it and then just start again. So some of the I would say critical steps for this is you want a well-stained tissue for this. So for example, roto, reduced osmium, TCH, second osmium. Embedded in EPON with hard formulation, the hard formulation is required for thin sectioning. Even if it's 60 nanometers, the hard formulation will prevent many of the artifacts that we'll see later on. Larger sections are prone to wrinkling artifacts. So in order to prevent that, it's important so if we were going to trim this block, we would want to make sure that the majority of that block face is tissue rather than empty resin. So that will help to reduce wrinkles. Now, the tape itself is not conductive, at least not that I know. Maybe there's new tapes these days, but there's two methods here that will significantly increase your success. One is glow discharging the tape. So you may be aware of glow discharging for TEM grids. You can create a reeling mechanism in your glow discharging chamber so that you can glow discharge the tape as it goes through. And that significantly helps with prevent the formation of wrinkles. And then second is carbon coating is essential for creating that conductivity within the SEM microscope. So you can either carbon coat the tape ahead of time and then collect the sections onto the carbon coated tape, or after it's been laid on wafers, carbon coat the whole wafer, and then it can go into the SEM chamber. So this is an example of what that looks like. So again, this is the snout of that tape collector. The tape is moving, so you can adjust the speed of the tape movement so that it either matches your microtomy speed. And hopefully you can see this, but as the sections are being cut, they're pushing the previously cut section onto the tape. And then in this case, the tape speed is just a little bit faster than the cutting speed in order to prevent contraction of those sections. So this is a schematic of that. So again, sections are collected up here into the collection reel. Then they're laid, so after this is collected, you can cut the tape onto sections, lay them on these 4-inch silicon wafers. So pretty big, they're different sizes, but these 4-inch silicon wafers are pretty nice. You can get stages specifically made for them that go into the SEM chambers. And then again, it's imaged with field emission SEMs, and detectors are getting better and better every year. So these days you can get beautiful images from them. And then the bottleneck, which is actually true for all of volume techniques, is that data alignment, data analysis, reconstructions, understanding what we're imaging is the next step. So a little bit of the pros and cons. What I think is the most important pro of this method is that it's non-destructive. So you can take your tissue, even if you're not sure exactly what you're looking for, as long as you collect the sections, they're presumably good indefinitely. I have a question mark here. It says wafers are stable indefinitely because I haven't tested this. We've only been working on this for 8 years or so, but so far it seems to hold on. So the sections can be also large. It really is limited to the width of your diamond knife blade. So 3 to 2 millimeters in diameter, that's pretty large. Then you can take those large sections and do low resolution mapping imaging runs. And that can help find rare events, rare cells that can be then specifically targeted for high resolution imaging. And again, since the wafers are presumably stable indefinitely, they can be imaged in the future with better detectors, which is what we're seeing today. And something that is also interesting is that the sections on tape are amenable to immunolabeling, and the second half of my talk is going to talk a little bit about that. Some of the cons, which are significant, is they're prone to artifact formation such as wrinkles or dust particles that fall onto the sections, and you'll actually see some of that in my videos. The alignment method can alter the data, so the sections are not always collected right in the same orientation. So there is a significant alignment that needs to happen after data acquisition. It is technically difficult, and expertise in ultramicrotomy is essential, especially if aiming for those 30 nanometer thick sections, which as I say, really significantly improve the data quality. And then lastly, it can be pricey, but I think all volume EM, all EM techniques can be pricey. In this case, it requires buying the commercial ATUMtome from RMC, which comes with the tape collector and the microtome itself. And then the frequent resharpening of diamond knives, depending on how much sectioning you're doing. So moving forward, all of my data is going to be showing ovarian follicles. And I'm not going to go into the biology in detail just for the sake of time and just because this is not super important right now. But just to introduce ovarian biology very quickly, in mammalian ovaries, oocytes or egg cells are found within follicles. So over here we have a cross-section of an oocyte, which is kind of like that, just at this angle. Then a lot of supporting cells around it. And these are all surrounded by a basal lamina layer around here, forming a spherical ovarian follicle structure. So in this case, we're going to be moving towards the center of that oocyte. And again, this is a low resolution, low magnification imaging run, which is why you see all of those artifacts. Typically, your high resolution data is unlikely that you will find so many artifacts in there. But I like showing this video because it highlights the volume size. So in this case, the oocyte is about 80 microns in diameter. So this is a pretty large field of view here, and this is just a crop from the entire imaging run. Then we can go ahead and start looking at the cells in more detail. And again, this was taken many years ago with an old Zeiss microscope. And we can get way better images these days. But again, this is 60 nanometer sections, and you can start to appreciate many organelles within it. Just a little bit of those ciliary bodies, mitochondria, ER, all of that beautiful stuff. And of course, reconstructions after the data has been aligned, we can start tracing them. and eventually, getting the 3D models that you're all probably used to seeing. In this case, this is just a single cell that's 2 or 3 layers away from the oocyte, and we're just looking at how that cell connects to the oocyte. And this is in context with its neighbors. The oocyte is down here, and the rest of the cells are up here. So this is after we upgraded to a better microscope and with a better detector. And what I wanted to show here is that 60 nanometer thick sections are enough for tracing thin cytoplasmic projections, which I think you'll be able to appreciate here. So what we're tracing here is just one of those cytoplasmic projections from the supporting cells up here, connecting to the oocyte through these thin cytoplasmic projections. And again, reconstructions can be acquired through that. Okay, so now to switch gears a little bit, this is what I think is kind of like the next level of the method, is kind of how to integrate it into immunolabeling. And I did some of this work back at UConn, but in order to show it to you, I'm just going to explain a little bit between the difference between pre-embedding versus post-embedding immunolabeling. So for pre-embedding immunogol, we start with a tissue. We fix impermeabilize, which breaks up those membranes so that the antibodies can go in. So antibody labeling is done in situ. But the advantage of it is that then that tissue can be processed for conventional EM and conventional ultramicrotome. So typically what you get here is pretty good antigenicity, especially if detergents are used, but morphology tends to not be as good. On the other hand, we have post-embedded immunolabeling, which is technically difficult, but possible. So typically you want to freeze your tissue if it's thin enough, embed it in an acrylic resin, section that, and then label the sections. So the problem here is that the antibodies can only bind to antigens that are exposed to on the surface of those sections. So antigenicity tends to be pretty low, specifically because of how few antigens are exposed to the sections, but morphology tends to be much better. So in order to explain, so I'm just going to very briefly explain a little bit of this biology here in order to show you some of the data coming through. But so oocytes, or egg cells in mammals, they are arrested in meiosis for the majority of their lifetime. This happens back in embryogenesis, and for the majority of their lifetime, they remain arrested in meiosis. And through some beautiful work done at the Jaffe lab over at UConn, what they've been able to show is that it's high levels of cyclic GMP in the oocyte that are maintaining that meiosis arrested. But it's the supporting cells, the granulosa cells, that are making that cyclic GMP. It's diffusing through gap junctions. So all of these cells are connected to each other through gap junctions, and these can get pretty large. In humans, they can get to up to 2 centimeters in diameter. So it's all connected. So then cyclic GMP diffuses through into the oocyte and keeps meiosis arrested. Now, prior to ovulation, the oocyte needs to resume meiosis in order to make it competent for fertilization if fertilization was to happen. And this happens from the signal luteinizing hormone, which is released from the brain, comes in through the blood, and then acts on particular cells. So what's interesting is that it's not the oocyte itself that senses the luteinizing hormone, and we know that from several experiments. We know that it's the supporting cells that are sensing the luteinizing hormone. Eventually, cyclic GMP production gets turned off. Cyclic GMP begins to diffuse out of the oocyte through the gap junctions, and meiosis resumes, and that begins the ovulatory process. So the question was, where is this LH receptor? Where is it expressed? At what point is it expressed? And all of that. So, okay, so LH receptor is a G protein coupled receptor. Maybe you know a little bit about this, but making antibodies against G protein coupled receptors can be challenging. It just wasn't possible. So what we ended up doing was tagging it with an HA tag and using commercially available antibodies to label that. So I know this is a busy slide, but I'll walk you through it. So what we're seeing here is pre-embedding immunogold on vibrotome slices. These are 70 micron thick vibrotome slices of ovaries, so cross sections of ovaries. What we're doing is we're labeling the HA-LH receptor with a commercial antibody and using a secondary antibody, which is nanogold conjugated with Alexa 488 conjugate from nanoprobes. And that's what we're seeing here. So on the left is that nanogold, that enhanced nanogold has been enhanced with a gold enhancement kit from EMS. So this purple stain here is that nanogold signal. And in green is the Alexa 488 conjugate. So connexin 43, this is just, I like using this as a positive control. This is the primary protein forming those gap junctions that I told you about. So the gap junctions are connecting all of these cells to each other. So it serves as a very nice positive control. And then no primary antibody as a negative control. So this is a great technique because at this point, you're only really worked on this for what, a day, maybe a day, and you left it overnight in antibodies. You don't have to wait until you process the whole thing for EM before getting results. So this is a great time to, kind of like a checkpoint. So at this point, it looks good. This signal looks good. This signal looks good. So now we can go ahead and process these vibrotome sections again, 70 micron thick vibrotome sections with full osmium, UA or roto, PO, and EPON embedment. So this is what we get after that. So this is those vibrotome sections. They have been thin, resting embedded, so kind of very thin, put on a slide, and then imaged by light microscopy. And again, what we see is that dark stain that is the nanogold that's been enhanced that is on those cells. And because this is embedded in EPON, we can take thin sections, put that on the tape, and start imaging with an SEM. We can correlate these signals to each other, and then target that specific ROI for high resolution imaging, which is where we expect to see a lot of that positive signal here. So what you're going to be seeing here is a mapping run, low resolution, 60 nanometer sections on tape of that immunolabeled section. And it looks like that because it's just very difficult to truly flat embed vibrotome sections after they've gone through that roto protocol. But what we can do here is look at this particular region of interest. And full disclosure, this is 0.1% Triton X, which is extremely high. So membranes are pretty much gone, but epitopes are there. And the reason why I wanted to use this high concentration is because it allows us to see kind of like these it's just a rough outline of where those cells are, whether the method is working, what needs to be improved. And so the nanogold is these small black dots, not to be confused with these larger ones, the spherical ones. These are lipid droplets, and they're all over the cells. It's the smaller ones. The signal is primarily lining the outer membrane of the cells and the ER within the cells. So this is just another view of that, where we can start to see some of those individual cells that are spread throughout the outside of the ovarian follicle, inside and outside. So then we can start to titrate down that Triton X and start to get better morphology. In this case, this is just a nice view of a positive cell next to some negative cells. And there is some background there, but it's significantly different from what we call a positive signal. And again, thanks to through the tape collector system, we can start to get some reconstructions and get something that looks like this. So it was actually surprising to see that there is heterogeneity in the expression of the LH receptor. We were not expecting this at all. We could see that here in green is cells that are expressing the receptor, and in blue cells that aren't, and they're right next to each other. We haven't yet discovered why some cells express it and some cells don't, but this was a good start. Something else we can do is, remember I told you the follicles are surrounded by a basal lamina where the blood vessels are here. The blood vessels carry the blood with luteinizing hormone, which is what eventually ends up triggering ovulation. And we can start to look at what those contacts look like. So again, positive cells, negative cells right next to each other. And these are those contact sites between those cells and the basal lamina. So we just started looking at that, some quantitation, no difference. No difference there, but just highlighting that is something that is possible, and just appreciate the size of that volume. This is many cells. I wish I had a scale bar here now that I think about it, but it's an extremely large volume. So if you happen to have a very nice antigen, such as connexin 43, which is the protein that is abundant in these gap junctions, you can get by with very small amounts of Triton. So it's a 0.01%. We can appreciate that the majority of the membranes are there. Morphology looks okay. And what we're seeing here is linear gap junctions, but also some internalized gap junctions over here, which I will touch on in just these upcoming slides. So it's just a reconstruction of two cells. In yellow are the gap junctions. And in orange is gap junctions between these particular two cells. So to end, I just wanted to highlight some of this work. This was done by Rachel Norris and Mark Terasaki. They've been working on this for several years, and I think the idea is brilliant. So what they're doing is, high pressure freezing tissues, embedding them in low acryl, cutting them with the ultramicrotome and collecting them on the tape. And what you can do is take that tape and cut it into sections, lay it on the wafer, and then label those pieces of tape with different antibodies. So what you can do here is, for example, if you have a cell that spans through all of these sections, you can, in theory, label them with all these different antibodies or a structure or whatever it is you're interested in. And it doesn't have to be this big. It could be, 2 sections, 1 antibody, 2 sections, another one. So it's quite brilliant. So Rachel wanted to use this method to study internalization of gap junctions, and this is something that happens quite a lot in the ovarian follicle. So this is what that looks like after a gap junction has been internalized. This is just an example of a gap junction that is going to be internalized after it reaches that cell. And this is not low acryl. It's just conventional EM. And there it is. So then she was able to label these gap junctions with several different antibodies. In this case, I'm only showing you one of the antibodies. It's just total connexin 43. What you're going to be seeing here is linear gap junctions on these 2 sides and internalized gap junctions. And the black dots are the gold label. So this is, low acryl sections on tape. I think they're 70 nanometers thick. And she's actually gotten much, much better at this. So this is a more recent paper where she's been able to see that these internalized gap junctions sometimes have entire mitochondria within them, multivesicular bodies, all sorts of structures. And this is a blow up of one of these panels. And what you can appreciate here is that inside the internalized gap junctions, there are connexin 43 positive membranes within it, which is quite interesting. So yeah, with that, I just would like to acknowledge everyone involved in this work. And again, this is work done at UConn. I'm no longer there, but remains close to my heart. So 2 big labs, the Terasaki lab and the Jaffe lab. Terasaki lab is kind of like the big EM lab, and the Jaffe lab is the big reproductive biology lab. So we all kind of work with each other. And of course, the UConn Health EM core, which actually they were the ones who taught me a lot of what I know about EM these days. If anyone wants to connect, please go ahead. I'd be happy to help and answer questions if anyone wants to continue with this method. I do a lot of FIPSM these days, so I'm also happy, though you have the true FIPSM expert here, but I'm also happy to help with that. Thank you.
Okay, yeah. Nice talk, thank you so much. Question. I used to do a bit of immunogold on section, and I'm intrigued by pre-embedding immunogold. So can you do, with the pre-embedding, you'll have much higher infiltration, and probably your labeling will be much more enhanced. Can you do dual size immunogold in that instance, or with so much enhancement this is going to be
yeah. So the question for the virtual audience is if it's possible to do dual labeling of pre-embedding immunogold, because what we're using is a nanogold that gets enhanced. I don't think you can do it in the same section. I'm trying to think about it, but what you can do, so in reality, the nanogold doesn't penetrate deeper than 20 microns. So that's why I went for 70 microns, which is about the easiest, because in vibratomes, you can't really go, I don't know, thinner than that, I think. But what you could do is label adjacent sections with different antibodies, if you're okay with losing a little bit of that. Because if you have a 70 micron thick section that is only labeled 20 microns on the top, 20 microns on the bottom, then your next section, your next slice will be a different antibody, 20 microns on the top, 20 microns on the bottom. That's the only way to think about it. I don't know if anyone actually has experience with that. So there's 2 different ways to enhance that nanogold, which is gold enhancement and silver enhancement. But I don't think you could differentiate. I don't think you could do the 2 without enhancing everything at once.
Basically just evens out. can we just do, because we are at 10, one last question. Yeah, 10.
So, first of all, like I found that for an envelope labeling, to use sapone because you can, it's a little bit gently around the tip.
Yeah.
And then it preserves the structure of the leaf.
Yeah, I agree. Mhm.
And then I've been using the color on the level, but I have a lot of problems with the enhancement using the gold enhancement kit. Like for none of them. So like, what are you?
Yeah, no, that's so interesting. So he is asking,
Oh, can you repeat the question?
yeah. So he's saying that he has some experience with the conjugated from nanoprobes to conjugated nanogold with fluorescence, and he's having problems with enhancement. I think I know what you're talking about. I had a lot of issues at the beginning at first because when you follow the EMS protocol, it doesn't work. Yeah, no, I know. Someone else actually discovered this and they told me, oh yeah, use tris buffer rather than water. I can send you, I mean, if you wanna,
Yeah.
or it's published.
Actually, Valentina, may I make a request on behalf of folks here, maybe even online, is some of your work is absolutely excellent on this, and this would be very useful. A very short protocol, if you can share
Yeah.
later on, just
Sure.
with little tips and tricks on this particular point. the
Gotcha.
pre-embedding.
Yeah. Because it was published as a research paper, but it really hasn't gotten a lot of citations or anything. But yeah, no, I'm happy to talk. Yeah.
Yeah.
Mhm.